Next up, we're going to have Hébert talking about neuronal replacement in the neocortex.
Hi. I'd like to thank U.S.A for inviting me, despite the fact that our projects in the field of regeneration are quite preliminary at this stage. So the brain, and especially the neocortex, presents unique challenges for regenerative medicine. When we look at a human brain, all we see pretty much is the neocortex and a bit of the cerebellum back here. And that's because our disproportionately large neocortex covers everything else. In fact, it's our most distinguishing anatomical feature compared to other mammals. And it's what we use to think, it's the seat of our consciousness, and to a large extent, it defines who we are. So if we're going to regenerate it, unlike for other tissues and organs, we not only have to maintain function in a general way, it's not enough to just have a working neocortex, we also have to preserve as much as possible the unique wiring and firing details that define us each individually. So we're focused on replacing the central type of neuron in the neocortex, the projection neurons. These neurons are glutamatergic, meaning they release the neurotransmitter glutamate. They're also the neurons that project to other parts of the CNS and to other parts of the neocortex itself. And they're shown here in blue. They're organized in layers in the neocortex. But just to put this in context, there are many other cell types in the neocortex as well, including the interneurons, which release the neurotransmitter GABA and which are essential for the normal development and function of projection neurons. There's also different types of glia, astrocytes, oligodendrocytes, microglia, and precursor cells, which are of interest if they can generate new neurons, as well as cells that are associated with the vasculature and the cerebrospinal fluid. So all these cell types will have to be considered to get overall cortical regeneration. But there's already a good evidence that these cells can be more readily turned over than the projection neurons. And since the projection neurons are really the central cell type in the neocortex, if we can replace those, I think we'll be in good shape. So is there any reason to think that we can even replace these neurons, even gradually over time, without disrupting function too much? Well, of course, there's going to be technical hurdles, but I think in principle it's possible. First, there's a lot of plasticity built into neural networks. This is a wiring diagram of some of the neurons in the worm C. elegans, much, much simpler than even a small part of the human neocortex. But the basic functional principles are conserved. Particularly relevant is the concept of Hopfield networks. A simple way of thinking about these networks is that given a particular input, even though a large number of neurons are activated momentarily, the activation pattern resolves to a stable state in which a subset of the neurons remain active for a longer period of time. When I say longer period of time, we're still talking like a second or a fraction of a second. And these stable states are called attractor states, and they represent memories. And depending on the input, different memories are activated. And these networks, is sort of simplified here, but are normally comprised of hundreds or thousands of neurons. And most importantly for us, they're highly fault tolerant. Whoops, sorry. Meaning they can sustain the gain or loss of some neurons. In fact, they need to be built this way in order to function. So this means that it might be possible to progressively replace neurons over time without causing too much chaos. So another issue is the sheer number of neurons that we'd want to replace, around 15 billion projection neurons in the neocortex. So for example, if we were to replace 100 million neurons a month for 15 years, we could replace them all. This may seem like a lot, but just for comparison, the adult mouse dentate gyrus functionally incorporates 15,000 new neurons every 6 months. And if you scale that up by volume for the human neocortex, that's the equivalent of 15 billion new neurons incorporated in 6 months, enough to replace all the projection neurons in 6 months. So obviously, speedy replacement can be counterproductive to maintaining our thoughts and our thought patterns and memories. But this is just to say that the numbers in themselves should not be a limiting factor. Right? But there are two major hurdles to replacing projection neurons in the neocortex. And this is where the work from my lab comes in. And again, I stress that this work is in its infancy and exploratory at this point. So the first is creating a receptive environment for new neurons. Because normally, endogenous precursors, even implanted precursors, cannot generate neurons that incorporate into the adult neocortex. The flip side of this would be to trick cells into behaving like they're in a receptive environment where they can develop and incorporate. The second major hurdle is the dispersal of neurons into the neocortex. Obviously, we can't go around implanting cells all over the neocortex. Right? So we need the cells to be able to do that on their own. So first, creating a receptive environment. And the first question we're asking is whether neuronal death in itself creates an environment that triggers the endogenous cells to replace the dying neurons. And certainly, if their neurons die due to stroke or trauma, they're not replaced. But it's hypothesized that this may be because of the necrosis and the strong accompanying inflammatory response. So maybe if neurons died by apoptosis without too much inflammation, then endogenous precursors would be activated to replace them. And Jeff Mackless's group in Boston tested this hypothesis about a decade ago and found that they could detect what looked like new neurons using markers for DNA synthesis, BRDU, and marker for mature neurons, NU-N. But these results have become controversial and are difficult to replicate and confirm. And one of the reasons they're difficult to confirm is that their approach involved 3 surgeries on the same animals, so it's difficult. But the question of whether or not endogenous precursors can be induced to produce new projection neurons in the cortex is still an important question. So we decided to take a transgenic approach, which involves no surgeries, to test whether or not neuronal death in itself can activate endogenous precursors to produce new neurons. And in fact, we've come up with 2 approaches that are successful in killing these neurons. In the first approach, we can activate an engineered caspase, which is a mediator of apoptotic cell death, specifically in projection neurons in the neocortex, simply by injecting a compound that causes individual caspase molecules to dimerize and become active, leading to neuronal death. And similarly, in the second approach, we can simply inject a compound, in this case tamoxifen, which causes the DNA recombinase CRE-R to translocate from the cytoplasm to the nucleus, where it recombines and activates expression of a diphtheria toxin, causing neurons to die. So both these approaches work. And this is fluorourate staining showing in bright green the dying neurons. And with the caspase approach, we see predominantly death in the posterior medial parts of the neocortex as well as the hippocampus, whereas with the diphtheria toxin, it's more evenly spread out throughout the neocortex. And in controls, we essentially don't see any cell death. So there are 2 big differences with what we see compared to what the Mackless group saw. So the first thing is we don't find much evidence for new neurons. We do detect what look like immature neurons co-labeled with the BRDU and the immature cortical marker double cortin. But this is not surprising since you can detect these even after stroke. But using confocal microscopy, we're really hard-pressed to find any new mature neurons that incorporate into the neocortex. The second difference, well, it also should be noted that even in the case in the Mackless study, the number of new neurons that they detected was far, far less than the number of neurons that they were killing. So the second difference is we can detect reactive astrocytes and microglia, signs of inflammation. Normally, the marker GFAP labels astrocytes only in the corpus callosum, not in the neocortex. So you don't see any GFAP labeling in the neocortex here. And microglia, although they're detectable with the marker IBA1, are non-proliferative. However, when we induce cell death, shown here in an adjacent section with fluorojade, we get activation of astrocytes, which now express high levels of GFAP, and the microglia become proliferative. As you can see, labeled yellow here, double label with the IBA1 marker as well as the DNA synthesis marker, BRDU. Right. So the Mackless group, although they never showed it in any of their papers, said that they did not detect any reactive cells. And this is surprising because even if we reduce the amount of cell death that we see by reducing the amount of inducers that we give to these animals, we can still detect reactive cells. Nevertheless, I think these experiments are useful in further establishing that just killing neurons is not sufficient to activate endogenous stem cells or precursor cells to give rise to significant numbers of new neurons. Although we haven't completely given up on the idea, and we're repeating these experiments now in the presence of anti-inflammatories, indomethacin and minocycline, to see if that helps. The second important point is even though these mice with the missing neurons in their neocortex don't trigger endogenous precursor cells to replace the dying neurons, they may provide a more favorable host for implanted cells to then give rise to neurons that can fill in these spaces left by the dead neurons. Which brings me to the second major hurdle. If we're going to use implanted cells, how do we get the neurons to disperse throughout the environment? And Arturo Alvarez-Bulla's lab, as well as other labs, have previously shown that if you take adult neural stem cells from the subventricular zone and transplant them in or around the neocortex, the neurons that are derived from those cells remain in a clump. Some of them go to the subventricular zone, of course, but none of them go disperse into the neocortex. And this is 30 days after implantation. The neurons are labeled in blue here. But there are neural precursor cells that are highly migratory. And these are the cells during development that derive from the medial ganglionic eminence. They give rise to neurons that migrate and populate the whole neocortex with the interneurons. These are the GABAergic neurons. Even when implanted in the adult, this is 1 day after implantation and 1 week after implantation, these precursor cells give rise to widely dispersed neurons. But the only problem is these are the interneurons, the GABAergic neurons, not the projection neurons that we want to produce. Okay. But we also know from developmental studies that there are factors that can transdifferentiate GABAergic cells into neurogenic, glutamatergic ones. For example, neurogenin-2, when introduced into these precursors in the ganglionic eminence, give rise to neurons that express glutamatergic markers, but not GABAergic markers. So our strategy for producing widely dispersed projection neurons in the neocortex is to harness the migratory potential of these precursor cells and then transdifferentiate the neurons that they give rise to from GABAergic to glutamatergic projection neurons. So we collect the MGE from embryos. This is medial ganglionic eminence. Dissociate the cells, infect them with lentiviruses with which we can induce expression of neurogenin-2 and other factors, and then inject these infected cells at the edges of the neocortex, wait a week or two for the cells to migrate, then hit them with doxycycline, which activates expression of neurogenin-2 or the other transcription factors, and then wait a few weeks to see what they become. And these are the lentiviruses that we're using, Neurogenin-2, as well as a couple of other transcription factors, PAX2 and EMX2, which are also known to, are able to transform GABAergic cells to a glutamatergic fate. And these are only expressed in the presence of doxycycline because of this promoter. And they also express GFP because that makes it easier to monitor expression of these transcription factors. And the cells that we infect, the MGE cell precursor cells that we infect, constitutively express the red fluorescent protein. So no matter what they become when we inject them into the mice, we can follow their fate. So, so far what we know from our culture studies is that MGE precursor cells, when infected with the virus and treated with doxycycline, express these transcription factors. So here are the infected cells in green, and their nuclei are labeled, they look yellow because they're co-labeled in red with PAX6 in this case. And here's a higher magnification, in this case with Neurogenin-2. You can see the green cells have Neurogenin-2 in their nuclei. And as expected, so far it appears that these cells are now acquiring a glutamatergic fate instead of a GABAergic fate. So in infected cells, we find the expression of the glutamatergic marker TBR1, whereas we rarely, if ever, find labeling of infected cells with GABA, for example. Right, so we're going to extend these findings in culture, and also we're now going to start injecting these cells into the edge of the neocortex to see whether or not they give rise to glutamatergic neurons after we hit them with doxycycline and after they've dispersed. Right. What we also need to do once we've done this is to check whether they give rise to the right subtype of projection neuron. I mentioned earlier that projection neurons are organized in layers, and each of the layers expresses different sets of molecular markers, and so we can check whether or not, based on their location, they're expressing the appropriate markers. If not, we can add another transcription factor. We have candidates for that to specify the layer, or the layer subtype of projection neuron that's there. We also need to check whether these neurons project to the right targets. Different layers project to different targets. And again, we can do this because we can see those cells are labeled red, and we can dye fill them with a very diffusible dye that will show us the axons so we know where they go, or we can use retrograde labeling as well from their targets. And also we want to test using electrophysiological approaches whether or not they're functionally incorporated. Right. But ultimately, the proof of principle that this can work would be to replace neurons in one area, like the visual cortex, where we know a memory is stored. For example, where a platform is in a simple water maze, it's always where the visual cue is, and see whether or not after we replace the neurons, whether the mouse can still remember where that platform is. And this is not an all or none situation. So intermediate successes would also be very valuable, right? Because that means that it's easier then to reacquire those memories. And even if we're successful in replacing glutamatergic projection neurons, there are still other components of the neocortex that would need to be regenerated in order to get overall rejuvenation. In particular, the GABAergic interneurons. If those are deficient, we're likely to develop seizures as well as other disorders like schizophrenia. So the balance between the two is very important to maintain that. Of course, there are other non-neuronal cells. The vasculature is an example. There's evidence that implanting or the presence of new neurons in glia can trigger some sprouting, but the bigger vessels will have to be dealt with. And of course, there's the extracellular matrix, which would have to be replaced or repaired. Again, putting in new cells, to some extent, they lay down new matrix, but the old matrix would still be a problem. But really, I think the biggest limiting factor right now is our knowledge of what works and what doesn't. And so we're very eager to start trying more of these approaches in vivo to seeing how far we can go with them. And with that, I'd just like to thank the people in the lab who've done the work, Frank Diaz, Nick McKeehan, and Christina Kratchmer. So,
Questions?
So what's the current state of the evidence that aging is accompanied by significant neural cell death? Have you had the rug pulled out from under you, or is this still regarded as something that needs to be repaired?
Right. No, I'm not saying that there's cell death with age, but certainly neurons become decrepit and do not function as well, right? And if you want to prolong your life to 150 and you're going to do nothing to rejuvenate these neurons, you're really not going to enjoy your extended life. I mean, you really need to rejuvenate those neurons.
Is there any hope of rejuvenating their synaptic complexes?
Right, so once they get there, that's one of the things we need to test, whether they become functionally incorporated, which would mean, yes, they would need to form.
No, but they wouldn't have your memories. They'd have.
Well, that's up in the air. It's possible that progressive replacement, you would get, you wouldn't necessarily lose your memories. Memory is a very dynamic thing to start with. So it may become more dynamic with the replacement of neurons, and to what extent, we'd like to test that, and we will eventually.
So your data with the taking the donor cells from the MGE suggests that it's not the intrinsic environment of the cortex which prevents migratory behavior of implanted cells, but rather it's the population of cells in particular that are being implanted.
Yes.
So have you thought about comparing the MGE cells with other populations of cells so that you wouldn't have to go through the process of getting them to differentiate along the lines that you wanted?
Well, I mean, a lot of work has been done with the more classic adult stem cells, like the subventricular zone cells, and they don't incorporate new neurons into the cortex. Do you have suggestions for other neural precursors? I mean, you can go during development and find a whole bunch as well, and people have done that actually, taken precursors for glutamatergic projection neurons and implanted them. And they'll migrate a little bit radially and incorporate, but they don't do very well either. They don't really disperse that much. But the good thing is, the ones that do locate within the cortical area, they do incorporate in the adult environment. So there may be a certain amount they can do on their own. So we won't have to program everything, just maybe getting the neurons there, and if they're of a glutamatergic fate, they may be able to do the rest on their own, which would be very nice.
Actually just opened a new window for me. The glutamatergic precursors don't migrate in the embryo either, right? They go
Short
just a relatively
differences.
short distance
Right.
radially, sometimes
Right.
a little bit tangentially, but
Not even
very
so
little
much, yeah.
relative to the NGE and the
Yeah.
LGE.
Correct.
Here. I think that your work is quite important, really. I encourage you to follow through. But the problem of memory, as pointed out, is an important one. Have you considered the possibility of using replacing neurons in systems that may be less difficult? For example, dopaminergic neurons in the nigra, where you need still the connections, but the system doesn't seem to be as complex as the brain, and apparently there are no memories involved there. So perhaps,
Yeah. No, I mean we're going for the going for the gold.
big
as it
prize.
were.
Okay. Thank you very much, Dr. Hébert.